Good morning, dear students. Welcome again to the new lectures of cell biology. As you know, the living things can classify them into the 6 kingdoms: Animalia, Plantae, Fungi, Protista, Archaeobacteria, then Eubacteria. The cell components are cell membrane, cytoplasm, nucleus, mitochondria, ribosome, endoplasmic reticulum, Golgi apparatus, lysosome, and some other components. About the cell membrane. Cell membrane are a crucial structure because they act as barrier between cell components and the external environment. The cell membrane is mainly composed of mixture of protein and fats, so that the cell membrane allow some molecules to enter the cells and prevent the others for that. Functions of the cell membrane, as I mentioned before, cell membrane have 2 essential functions: keep the toxic substances outside the cell. It contains a receptor and canals that allow specific molecules such as ions, nutrients, metabolic products to enter the cell, so that, as I said before, allow some molecules to enter and to prevent the others to enter the cell. About the cytoplasm. All cells can be divided into 2 major components: the nucleus and cytoplasm. The cytoplasm is the region outside the nucleus. It is divided into deep endoplasm, and this is surrounding the nucleus, and the other part, thin superficial ectoplasm, just beneath the cell membrane or under the cell membrane or plasma membrane. In cell biology, the cytoplasm is a highly gel-like. Their structure is like gel. Gel-like substances or substance enclosed within the cell membrane. Functions of cytoplasm regulates exchange of materials out of the cell. Regulates this exchanging. Also, inside the cytoplasm, biosynthesis of macromolecules, big molecules. Also, movement of cell components inside the cytoplasm. 3. The nucleus. In biology, the nucleus is a specialized structure found in most cells, except bacteria and blue-green algae don't have mitochondria. It is separated from the rest of the other cell components and covered by a double layer called nuclear membrane, so that it is covered with the nuclear membrane. The nucleus controls and regulates the cell's activity, like growth, metabolism, etc., and carries the genes and structures that contain the hereditary information of each organism. The main components of nucleus are phospholipid bilayer membrane, nucleoplasm or nucleoplasm, nucleolus, chromatin. Nucleolus in Arabic, النوية. So that we have here, this part is nucleus, and they have this is nuclear membrane, envelope the same, and nucleolus inside, and here neuroplasm between the nucleolus and the membrane called nucleoloplasm. And here we can see here pores to enter molecules inside the nucleus, and also to enter like messenger RNA. Phospholipid, so that a phospholipid bilayer membrane, nucleoplasm, nucleolus, and chromatin. The main functions of the nucleus, first one, or important one, a protein synthesis, also cell division and differentiation, cell type. We have a lot of types of the cell, so that it's responsible for the differentiation of the cell type, control the synthesis of enzyme involved in cellular metabolism, controlling heredity traits، يعني الخواص أو السمات الوراثية of the organism, store DNA strands, also store protein and RNA. Mitochondria. Mitochondria, you can saw this picture or see this picture. Inner membrane, out membrane. We have this is inner membrane, and this is outer membrane, and some structures called the cristae and matrix. Mitochondria are an organelle not found in all cells. They are present in almost cells of eukaryotic, like animals, plant, fungi, and protista, but are absent in all prokaryotic, like bacteria, archaeobacteria. Biochemical process of respiration and energy production, so that synthesis, or they synthesize the ATP, have occurred inside the mitochondria. It has double membrane. The mitochondria have double membrane. The inner membrane being folded يعني تحدث لها طيات، يعني يشتيق طبقة وربيتشوني inward إلى الداخل to form layers، تكون ما تعرف ب cristae, and consists of the following: outer membrane covers the organelles. The inner membrane is composed of phosphobilayers with protein. In this picture, we can see the ribose and deoxyribose. The both it's pentose, and the carbon atom. This is B number 1. This is 2, 3, 4, 5. So that have 5 carbons, and due to that, it's called pentose. 1, 2, 3, 4, 5. The ribose and deoxyribose, the differences between them: in the deoxyribose, we don't have OH. The ribosome are complex structure of RNA and protein that carry out protein synthesis. It comprises 2 sections known as subunits. The smaller subunit is the place where the messenger RNA bind, and the bigger subunits is the place where the forming and bringing amino acids. So that in this picture we can see here big subunit and small subunit. messenger RNA bind with the small subunit, and about the amino acids bring by the tRNA to produce the peptide or the protein. In this picture, we have differences between eukaryotic and prokaryotic. You can see the ribosome in the prokaryotic called 70S, in eukaryotic called 80S, and consisting of two subunits each one. The subunit 50S and the subunit 30S. For the eukaryotic, the 60 subunit, the name of it, and 40S subunit. The prokaryotic have two genes here. In the bigger subunit for the prokaryotic, 5S RNA, 23S RNA. For the small subunit, 16S RNA. About the eukaryotic, also have here we can see three genes, 5S RNA, 5.8S gene, and the others 28S RNA. All of these three inside the bigger subunit. For the small subunit, 80S RNA gene. Six, the endoplasmic reticulum, ER. It is a network of tubules, smooth endoplasmic reticulum and flattened sac, rough endoplasmic reticulum, that serve as variety of functions in cells. The two regions of the endoplasmic reticulum different in both structures and function. So that we can see here, this is like a tube, a smooth endoplasmic reticulum, and flattened sac here around the nucleus called rough endoplasmic reticulum. Now, the differences between rough endoplasmic reticulum and the smooth endoplasmic reticulum. For the rough has a ribosome attached to the cytoplasm site of the membrane, so that the ribosome attached to the rough endoplasmic reticulum from the site of the cytoplasm, not in the site of the cell. It is a series of like flattened sacs، يعني مثل أكياس مفلطحة. For the smooth endoplasmic reticulum, like ribosome, don't have ribosome here. The smooth ribosome, or the smooth endoplasmic reticulum, sorry, it is a tubule, like الأنابيب, tubule network. About the rough endoplasmic reticulum, the ribosome attached to the rough endoplasmic reticulum synthesize protein، تصنع البروتينات by process of translation. Translation يعني messenger RNA هي التي تأخذ نسخة من هذا البروتين أو الذي يراد تصنيعه وتذهب إلى السيتوبلازم إلى الريبوسوم. Smooth endoplasmic reticulum is responsible for the synthesis of essential lipids such as phospholipid derivatives, cholesterol, steroid hormones in the cell. This is all these chemicals synthesized inside the smooth endoplasmic reticulum. Here I want to see this example. This is dichloroethane. Dichloroethane, we can see here C2H2Cl2, but their structures must be different. And when we see the chlor, the two atoms of chlor in one side, at that time it is called cis. If have each atom chlor, each one in the different side, at that time it's called trans. So that dichloroethane C2H2Cl2, it is an example of cis and trans opposite side. Here, Golgi, this is cis phase, meaning that all the opening of the endoplasmic reticulum, rough and smooth, it be in the one side, so that it is called cis phase. Here, incoming transport vesicle يعني حويصلات تدخل يعني أو قطرات تدخل داخل الGolgi apparatus. This is also the same. And lumen تجويف. About the transphase, it be opposite transphase. Here, these vesicles is outgoing, يعني استقبلت وعدلت وخرجت من الGolgi apparatus. Golgi apparatus, it is composed of a flat six يعني من أكياس مفلطحة known as Christina, E silent, Christina, which are stacked and semi-circular around the nucleus. تتكون من أكياس متراصة وشبه دائرية حول النيوكليوس. The multiple sake of the Golgi have different chambers for chemical reaction. فهذه الأكياس تحتوي على حجرات يتم فيها التفاعلات الكيميائية which allow them to increase the speed of protein production إنشاء أو تكوين البروتين. 8. Lysosome. Lysosome are discovered by the cytologist Christian Rey in the 1950s. A lysosome is a subcellular organelle that it is found in all types of eukaryotic cells. It is responsible for digestion of macromolecules such as nucleic acid proteins, polysaccharides, alt cell parts, and microorganisms. We can hear the lysosome found in the eukaryotic and their functions, mainly digestion of macromolecules like nucleic acid, proteins, polysaccharides, all part of the cells and microorganisms. In this slide, we can see the animal cell, and this is contain all main components that I talked about before. Thank you for your attention. And thank you for your listening.